Hello, my name is Salomé Pinho. I'm a senior researcher and leader of the Immunology, Cancer and Glycomedicine group at the Institute for Research and Innovation in Health in University of Porto, Portugal. My work focuses on immunology and cancer, particularly on how protein glycosylation modulates essential cellular and molecular mechanisms in immune-mediated diseases and cancer. Animal experimentation is an important component of my research in my lab, and therefore I'm particularly sensitive to animal welfare and well-being issues. Next Thursday, I will be answering your questions concerning animals as essential preclinical models before a first-in-human clinical trial. See you soon! So depending on your scientific question and the need to use complex systems as animal, you should choose the best animal model that mimic the desired biological context to make your questions and respecting the 3 R's principle. So this always depends on the disease that you are studying. There are animal models that closely resemble differences in age and sex in terms of disease incidence and pathogenesis, but there are other diseases that can be modeled as well in animals, but with little or no sex or age bias, and therefore not perfectly translating or mirroring what is observed in humans. So animals are essential preclinical platforms, mimicking a complex biological system to test an hypothesis, such as a mechanism or a drug, before a first-in-human trial. This is particularly important when we are studying the interplay with the immune system, such as in cancer and inflammatory diseases, in which animals are actually essential models. So depending on the question that you have, you should always look to alternatives such as invertebrate species or other vertebrate models, respecting always the 3 R's principle. Although there are scientific contexts and questions in which mice are the only mammalian species suitable to study as a way of more precisely translating the evidences to human patients. This is particularly relevant when you are studying, for example, the immune system. Yes, indeed. We are studying other inflammatory diseases, and of course the knowledge and know-how that we are using in these other immune-mediated diseases, we are also applying to a project that has been running in the lab in COVID-19. So taking into account the fact that the research focus of my lab is in cancer and in inflammatory diseases, I mostly use mouse models, envisioning the translational application to human patients. Why? Because although some invertebrate species offers the power of genetic manipulation, the immune system is completely different, and this of course would constitute a big limitation to investigate our hypotheses, predominantly when we are talking about the immune system.